Hey there, science fans! Today we're diving into the fascinating world of molecular biology to explore the central dogma. Think of it as the fundamental principle governing how information flows within our cells, guiding the creation of you and me from a single cell. It's like a blueprint, but instead of buildings, it builds life itself. The central dogma outlines the intricate processes of DNA replication, transcription, and translation. These steps describe how genetic information stored in our DNA is transformed into functional molecules that drive every aspect of our existence. It's a bit like a recipe book, with DNA holding the master recipes, RNA acting as the messenger, and proteins as the delicious dishes. Understanding the central dogma is crucial for unraveling the mysteries of life. It helps us understand how we inherit traits, how our cells function, and even how diseases develop. So, buckle up as we embark on this incredible journey into the heart of our cells. DNA, or deoxyribonucleic acid, is like the celebrity guest of the molecular world, and for good reason. It's the molecule that carries the genetic instructions for building and operating an organism. Think of it as the ultimate instruction manual, passed down from generation to generation. DNA is structured like a twisted ladder known as a double helix. The rungs of this ladder are made of pairs of nitrogenous bases: adenine, A, thymine, T, guanine, G, and cytosine, C. These bases pair up in a specific way, A always with T and G always with C. This specific pairing is crucial for DNA replication and the accurate transmission of genetic information. The sequence of these bases along the DNA strand forms the genetic code. It's like a language where different combinations of letters create different words, and in this case, different genes. Next up, we have RNA, or ribonucleic acid, a close relative of DNA. RNA is like the messenger that carries the genetic instructions from DNA in the nucleus to the protein-making factories of the cell, called ribosomes. Unlike DNA's double-stranded structure, RNA is single-stranded. It also uses a slightly different set of bases: adenine, A, uracil, U, guanine, G, and cytosine, C. Notice that uracil U replaces thymine, T, in RNA. There are different types of RNA, each with a specific job. Messenger RNA, the mRNA, carries the genetic code from DNA to the ribosomes. Ribosomal RNA forms part of the ribosome structure. Transfer RNA, tRNA, brings the correct amino acids to the ribosomes to build proteins. Think of them as a team, working together to translate DNA's instructions. And finally, we arrive at the proteins, the workhorses of the cell. Proteins are large, complex molecules that carry out a vast array of functions in our bodies. They are involved in everything from building and repairing tissues to transporting molecules and fighting off infections. Proteins are made up of building blocks called amino acids. There are 20 different amino acids, and their specific order determines the protein's unique three-dimensional shape and function. It's like building with Legos. Different combinations create different structures. The genetic code carried by mRNA dictates the precise sequence of amino acids in a protein. This sequence determines how the protein folds and ultimately its function. So DNA holds the blueprint, RNA delivers the message, and proteins are the final product.